Good afternoon. I'm Heather Erickson, President and CEO of the Life Sciences Foundation, and I'm thrilled to welcome you to this event today, the Centaur and the Whale, Cetus-Chiron, and the Emergence of Biotech, and really the Emergence of Biotech both here in the Bay Area and around the country. First, I'd like to extend my sincere thanks and appreciation to our panelists. We have an incredibly distinguished group of folks who have come together, in many cases not from nearby, to join us today. And they'll be discussing the topic at hand and hopefully providing a little bit of insight. Also, our partners in promoting this event, Bay Bio, UCSF, and QB3, as well as hosting us here on the campus. And in particular, my special thanks to Ed Penhoet, Hollings Renton, and Bill Rutter, who have hosted today's program and made it free and available to all of you. I've enjoyed learning and hearing from them over the past several months, and their enthusiasm for this subject has been contagious, and I'm looking forward to more of that discussion here with us today. Briefly, by a show of hands, how many of you are alumni from either Cetus or Chiron? Wow. Well, I should raise my hand as well. I actually had the pleasure of interning for 2 summers at Chiron in the 90s in Rusty Williams' genomics lab. So I can attest to just how special a place it was and just how special it is to be in that type of environment. So hopefully we'll have a chance to celebrate that a bit this afternoon. I'm also heartened to see a lot of members from the community here. We've had a lot of interest from students and faculty, and a good number that will probably be rolling in the next few minutes. But really a chance to learn from the folks who blazed the trail that we're now building the industry on. Much has changed in many ways from the 1970s and 80s when these companies were plotting that initial course. But I think we'll also hear a bit about how we can learn from and continue to take some of those initial steps forward from the successes that Cetus and Chiron had in those early days and how they apply 30, in some cases 40 years later. So that's a lot of why the Life Sciences Foundation exists. We're here to capture the history, to preserve the heritage, and to tell the stories of biotechnology, to provide continuity from generation to generation, and to use that as a powerful tool for education and engagement. It's ultimately about making sure that in collecting that, we're engaging the next generation, we're inspiring future entrepreneurs, we're making the science and what many of you have dedicated your lives and careers to accessible and relevant to the many people who are impacted by these discoveries and therapies every day. There's certainly an urgency to this mission. We only have a limited opportunity to record any one person's story, and the lessons from the pioneers are an important part of that. The witness of the formation of what will be probably the defining industry for the 21st century is critical, and now is our opportunity to really get that story firsthand from the people who made it. So, I'd like to take a minute and just recognize there are people like Don Glazer, like Peter Farley, some of the founders of companies like Cetus that aren't with us anymore. And we need to be able to recognize, remember the contributions they've had, and make sure that that legacy and that information is available for future generations. So for this reason, I'm thrilled that we have such an inspiring and knowledgeable group of leaders here with you today. This all-star lineup is not something that is easy to pull together, but we hope to be able to do it on many times again in the future with other leaders and other companies. And for the program itself, I'd encourage you to look at the printed program to get short bios on the 2 companies. Many of you may know these. There might be some snippets in there that you hadn't realized, and also brief bios on our individual speakers. We're not going to focus too much on the comprehensive history, but instead, what are some of those really compelling lessons that we can learn today? And I encourage you to start thinking about good questions you can ask these individuals, either your former colleagues or in some cases colleagues you would aspire to have, so we can really put them on the spot at the end of the program. So now I'd like to invite Steve Burrell, who is the CEO of Burrell & Company and also chairman of LSF's board of directors, to introduce our panel.
And there was a certain kind of East Coast Mafia story, and there was a West Coast Mafia story, and I was interested that we at least get the real story out. So we created in my office this foundation for the sole purpose, as Heather said, to gather up while we could the stories of the birth of this industry, the story of the growth, the story of the leaders, and what actually made it all happen. So some 3 years ago we got started. Arnold Thackeray and I were the initial leadership of this. We recruited a board and some of my friends, and off we were in building what is now a sizable foundation. We thank you for your support for all of that. Some of you may or may not know that in 1969, actually 2 years before the formal beginning of Cetus, we actually formed a partnership that brought Pete and Ron and some other people together, and Cetus was formed 2 years later as a corporation, 1971. And I had the pleasure, I was with Ernst & Young at the time, or Arthur Young at the time, to be involved in the start of Alzin, Cetus, and Genentech, and Amgen, many of the companies that became great companies today. So it gives me enormous pleasure to introduce a bunch of my old friends. This is the family that made biotech happen. I think Genentech takes a lot of credit for being the birth of biotech, but both of these companies, Cetus formed in 71 officially, Genentech in 76, Chiron in 81. So we had an industry being born literally right here. And as you know, a lot of it spun out of Bill Rutter's lab in the biochemistry department here at UCSF in the early 70s. So we owe an enormous amount of gratitude. I'm going to ask my friend Ed Penhoet to come up. Ed's going to moderate this discussion. And the discussion includes Hollings Renton, who, as many of you know, was president of Cetus for a while, came to Cetus about 10 years after it was formed, but ended up in leadership. Frank McCormick, very involved in the science that drove that company. Bill Rutter, of course. Pablo Valenzuela, one of the scientific leaders at Chiron. And so we've got quite a crowd of people. And so if you would join them on stage, we'll get started.
You know, fact is, I'm still alive, so I'm glad to be here, and I'm sure the rest of my colleagues share the same sentiment. It's wonderful to see all of you here. Thank you so much for spending part of your day with us today. We'll try to make it as useful as possible for you. We sort of divided this into 3 sections. One is to first talk about Cetus. It antedated Chiron by 10 years, as you just heard. Then we'll talk about Chiron, and then we'll talk a little bit at the end about the merged companies that came together in the late 80s, early 90s to form the merged company. Just a note about what these companies have accomplished individually and together. In the case of Cetus, best known for the discovery and the practical use of PCR, and Tom White played a crucial role in all of that, and Tom's here to describe some of that and other aspects of the company. Interleukin-2, which had a checkered history, but to this day is still one of the very few molecules which will actually cure a cancer. And now that all this interest in immunotherapy has emerged, a key molecule in the beginning of that field. Beta-Chiron, which was developed within Cetus, and today Bud Colby reminded me it's the 20th anniversary, not specifically today, but this year, 20th anniversary of the approval of Beta-Chiron. It's helped hundreds of thousands of people with multiple sclerosis. Something people don't remember, but maybe you do, Steve. AgroCetus is actually the foundation of most of what is currently Monsanto's plant effort. It evolved into a number of different things, but started within Cetus. And then Frank McCormick developed first at Cetus, passed through Chiron briefly, and then on to Onyx. Frank was the first, I think, to recognize the value of molecular oncology and developed the first kinase inhibitor that's widely used in the cancer field. In the case of Chiron, the development of the hepatitis B vaccine was really the first recombinant vaccine, came early in the company's development. The discovery of hepatitis C, of course, transformed blood screening and diagnosis of this terrible disease. The invention of viral load testing occurred to some degree, mostly at Chiron, because we were developing something called branch DNA, which was inherently quantitative in its approach, so you could actually test how much virus was present in the patient sample. But Cetus played a key role in the use of quantitative PCR later in measuring viral load. Everybody thought we were crazy to waste our money on this, but now it's, of course, a key part of every diagnostic procedure for many infectious disease. The production of insulin and yeast, about 2/3 of the world's insulin today is made using the alpha factor system, which was built by Pablo and his colleagues at Chiron, together with a number of academic collaborators, Ira Herskowitz from UCSF, Jeremy Thorner from Berkeley, Randy Sheckman, et cetera, all skilled in the yeast area. Commercialized a product for wound healing called Regranex. Actually did a lot of pioneering work on factor VIII and was, believe it or not, the first to clone any part of human factor VIII, and has now licensed several of the current producers. So in aggregate, these two companies, I think, have had an enormous impact on medicine, and we're here to sort of describe how those things came about. So we're going to start with Cetus. Cetus was started in '71 as a company to develop, using technology Don Glaser had developed in his lab, to develop a system for actually screening microorganisms to get the best producers in any system. But in the late '70s, morphed into a molecular biology company, and Tom White was a key person there. Tom, maybe you can describe a bit of how that transformation occurred from Cetus as a company doing enhancement of selection of microorganisms to the recombinant DNA world that started when you joined basically in the late '70s.
Well, I joined in July of 1978, and at the time, Cetus had two different divisions. The chemistry division, which did some of the screening for improved antibiotic biosynthesis for sharing. I think it was gentamicin was a drug, and for Russell cloth that had a program to increase the production of vitamin B12. David Gelfand, who's here in the audience, had started the recombinant molecular research department in 1976, and after that, there were several projects, not only the cloning projects on interferon beta with a mutation that allowed it to fold correctly in bacterial production, unlike the native interferon, which misfolded due to odd number of sulfhydryl groups, but also had projects that at the time were way ahead of their time, because Weyerhaeuser and National Distillers had projects to look at the cloning of amylases that could be used to produce ethanol from starch, and also to degrade cellulose waste into glucose and ethanol from paper pulp waste, things that are now of more considerable interest as a biofuels kind of approach. So this was done really almost 30, 40 years ago. And those projects actually were successful, but the companies themselves never commercialized. But with regard to the production of the human proteins as biologicals, the real question in that 78 to 85 period, which ones Cetus could work on that would be competitive with the companies that had already started factor VIII or interferon alpha, et cetera. And so the mutant to beta-Chiron was thought to give a time advantage for production, clinical trials, and approval. But most of the emphasis was on interleukin-2, which failed as a broad anti-cancer drug, but was useful for renal cell carcinoma. But other drugs, other biologicals that had a logical reason to be successful, like tumor necrosis factor, as a cancer treatment, failed utterly and were terribly toxic. But inhibitors of tumor necrosis factor became the treatment of choice for autoimmune disease and rheumatoid arthritis, just the opposite of what we thought originally. And macrophage-specific colony stimulating factor was also produced. It was a failure in the clinic, and only granulocyte colony stimulating factor and GM colony stimulating factor became major drugs, of course, for Amgen. So there was a good reason to do some things, but a number of them failed as drugs, even though they made sense initially as biologicals.
Mm-hmm. That was an important part of the early industries. Companies like Cetus and Amgen actually had a whole diverse set of things that were being pursued essentially because you could do them with the technology, and then over the years focused more and more sharply on human health applications. So
yes, especially with the first public offering in '81, which raised $108 million, the largest corporate public offering at the time. And that really then transitioned the company into focusing on recombinant DNA and biologicals.
So Frank, you joined somewhat later and really started the molecular oncology program within Cetus. And,
yes.
And since then, you developed the fundamental technology of looking at kinases as targets for drug development. And then you went on to actually get one of these approved when you were at Onyx. Wonderful job. Hollings deserve credit for, but probably the Onyx story is for another day. But if you could talk a little bit about...
Oh, come on.
I am the moderator after all. If you could talk a little bit about how you came to that. And then, Frank, the other interesting thing is, to me, you were able to establish a pipeline at Cetus, but at the same time establish yourself as a world leader in the science of kinases and their role in cancer. So can you talk a little bit about how you blended those, were able to do both of those things?
Okay. Well, I had several roles at Cetus. I was overseeing various research projects, trying to find new targets and develop therapies, but my particular focus was on using the new information that was coming out that showed that mutant proteins cause cancer. And that information started coming out in 1981, 82. And so I thought that would be an opportunity to develop diagnostics based on the mutations to detect cancer, and also to use the mutant proteins as targets. But it was pretty early on in the whole process of trying to develop drugs. So I proposed the oncogene project, and I think Tom gave me the thumbs up for the project, but I was also required to raise funds from the outside to reduce the risk. So we did a collaboration with Hoffman and Roche to do a lot of the screening, and got a grant from the NCI, the National Cooperative Drug Discovery Group, to help us fund that. So we sort of shared the risk of a very early project by getting funding from other sources. And that enabled us to interact with academics and keep a very high profile in the sort of academic world of publishing papers, but also moving along the agenda of drug discovery. The diagnostics part, we tried to make antibodies against mutant oncoproteins as a diagnostic tool, but then PCR came along in the lab next door to mine, so that approach was blown away pretty quickly, and we focused on PCR instead. But really, I think it was to Tom and Hollings and others' credit that they allowed me to do really basic stuff by sharing the cost and the risk with others, but also required me to oversee a bunch of other projects which were more focused and more applied. So it was a good mix, I thought.
A lot of similarities between the approach that Cetus and Chiron took during that era of leveraging the research.
Yeah, we were able to use technology, the cloning technology, not just for making recombinant proteins as potential drugs, but also making proteins for assays and modifying proteins to be suitable for assays using epitope tags, for example, which we were, I think, some of the first to use tags to purify proteins quickly, and baculovirus systems to make recombinant proteins for biochemical assays, which really enabled us to do sort of biology in a sort of high throughput, systematic way, which gave us a bit of a competitive edge.
Well, we've come a long way since you and I were both trying to figure out how to make a monoclonal antibody on the second floor of the biochemistry building at Berkeley. So, Tom, that sort of leads us right into a discussion of PCR. So there's a lot of lore about PCR, but you were there, you saw it all happen, and you made a lot of it happen. Can you give us a little perspective on the science of PCR, how it really evolved, and what the sort of key discoveries are? And I know David Gelfand played a very important role in a number of aspects of this as well. So,
yeah. Well, I think it's absolutely clear, of course, that the concept of PCR was Kary Mullis's, for which he won the Japan Prize and the Nobel Prize. But I think the two people who really contributed to getting the concept actually to work experimentally with data that could convince not only me and some of the other key scientists that it actually worked, were obtained by two other people in the audience here. One is Randy Saiki, who was the first author on the first paper that described PCR. He actually got the data that convinced me that it worked. And Randy made a number of other important contributions to the method. He's the first one who got the Taq polymerase conditions to work, which transformed, of course, the ability to have an enzyme that would survive the process rather than be added at every step. And Randy was also crucial to the invention of the TaqMan assay, which became the quantitative version of PCR that was standard to it. And Henry Ehrlich was one of the few molecular biologists, I think, along with Frank and myself to some degree, who actually thought Mullis's idea would work. Almost everybody else thought it would never work. For a variety of good reasons at the time.
That we don't have to go into here.
Which we won't,
Make it work for himself.
which we won't go into, but I think the crucial controls that needed to be done were really proposed by Henry. Randy was working for Henry, and he and his laboratory did really the first applications to using PCR for diagnosis of genetic diseases, sickle cell anemia, and then all forensic methods today are really based on the work that was initiated in Henry Ehrlich's lab. So it was a team effort that was really crucial, and I think without those key contributions from other people within the milieu of Cetus that was partially focused on commercialization and also focused on the explosion of potential applications that actually developed it to the extent that it's used today.
Well, Hollings, can you tell us a little bit about how the business of PCR evolved and devolved, so to speak? And
Well, it got away from us, I'd say, in short, sort of. In the end, we ended up selling it to Roche for $330 million, I think, plus some royalties. But clearly, we didn't, I think, really fully recognize for the Cetus shareholders the value of PCR. And I think, frankly, nobody in the company really appreciated it. I think Jeff Price, I don't know if Jeff's here today, but he recognized that maybe there were so many labs that there would be quite a few instruments and reagents, but it wasn't going to be as meaningful as it turned out to be. Brian Atwood, I think, was a big champion for getting out there and maybe doing something on our own. But in the end, we were basically a fully integrated pharmaceutical company. That's what we were attempting to do. The vision was very clear. It drove us to how we allocated our capital, which was there. And so when the PCR invention happened, we had people like, some Nobel laureates saying you should just make it available to everybody. And we said, no, we're going to commercialize it. But since we're putting all of our capital into the pharmaceutical side, largely IL-2 and some other proteins, we didn't have the capital to do it on our own, nor the expertise. We really had other expertise within the company to get it commercialized. So we went out looking for partners, and in the end, when we first went out, not very many people appreciated it either. We ended up doing 2 deals with people you probably wouldn't have expected, PerkinElmer, as opposed to some larger, maybe instrument company, and Kodak, which wasn't in diagnostics at all at the time. After 3 years, it was pretty clear Kodak wasn't going to do anything with it. We took it back. And I think that was probably the one opportunity, maybe when I look back and say, maybe could we have done something differently? Maybe we might have held on to it if we had have partnered more of the pharmaceutical stuff off, changed the vision, really. We'd had to change the vision. Yeah. And you don't change visions very often in a company. You had to change the vision and shared the risk in the pharmaceutical side of things and maybe taken a little bit more investment on our own in PCR, kept it longer, and maybe ended up trying to do a 50-50 deal as opposed to licensing it off. And then the rest is kind of history, and we'll kind of get to that maybe when we lead up to the merger there.
Yeah, I think I could add a little something to that because I'd actually joined Roche in '89, and all the rights were acquired in '91. But in 1991, the total sales of Taq polymerase, which was the only thing being sold for PCR, were 20 million. So Roche acquired all the PCR rights for 330 million. And the question, as I reflect back on it, is suppose Cetus had held back the research rights and sold Taq polymerase itself, which quickly became a $100 million a year market just for that alone. That could have been very profitable, but then Roche never would have paid the 330 million for it. So would that have allowed Chiron then to buy it at a lower, at a lower valuation? But like Hollings said, Cetus didn't have the experience in international regulatory affairs, commercialization, the customer thing in diagnostics. That really, I think, only could have been done by Roche or a comparable partner.
Mm-hmm. Well, we ended up partnering the hepatitis C assets at Chiron for largely the same reason. It would have been very difficult to penetrate all this market. And in retrospect, the decision to focus on oncology might not have been the wrong decision. I mean,
yeah, well, no,
it didn't
I
play
actually
out,
think
but
we
had
had
assets in.
Yeah. I mean, we did end up negotiating a good deal at the time. Mm-hmm. It seemed like a good deal at the time. Right. We clearly underpriced it. We actually had this thing. We had a very transparent process with Roche where we shared our sales projections, which were as high as we could possibly make them, we thought. And they had theirs. And then we said, but if they're higher, we'll sell it to you for a certain price, but we want what we called a blue sky royalty. And so I think Chiron was the recipient of some of those blue sky royalty payments ultimately. But,
and the motivation for Roche actually was to buy back the royalty stream because they couldn't really figure out what the potential would be. But once the HIV test became quantitative, PCR diagnostics became the most profitable product at Hoffmann La Roche in its history, more than Valium. I mean, it's kind of astonishing that was the case. So.
Well, it was a nice was a nice synergy between Chiron and Cetus because we had a lot of the IP on the HIV genome, which was necessary for those same assays. So both companies got
Right.
benefit from the Roche commercialization. But, you know, back to this issue of the choice to focus on oncology. I mean, at the time, in addition to the beautiful work that you've done over the years in the kinase field, Cetus also had programs in, some nice programs in the monoclonal antibody space, David Ring's programs, among others that I remember, and some of the immunoconjugates that were also the bispecific antibodies, I think were, if I remember correctly, there was one that had IL-2 on one side and a T-cell target on the other. So those things, you know, could have developed nicely in a different.
Right, maybe we're a little too early in that field. We had a whole immunotoxin program and a therapeutic monoclonal antibody program and a lot of things which were the right idea, and now are obviously the foundations of many other companies. But at the time, they weren't in a position to really bear fruit. But, So we decided on focusing on oncology to sort of bring together all these different technological assets into one disease focus. We had sepsis, inflammation, and many other areas we're interested in early on.
So maybe we'll switch to the Chiron side of things, and maybe Bill, start with you. Clearly you were a pioneer in many aspects of that was, you know, probably the most important hotbed, if you will, of the development of recombinant DNA technology in many different ways. But somewhere along in there, you became interested in the commercial side of things and decided to start a company. So what led up to that? And then clearly, as you'll describe, a key part of this was to develop a vaccine with the technology. And I know you had some very interesting thoughts about that going forward. Can you share a little bit of the background? Why start Chiron?
Well, I came to UCSF with the dream of having a science department work effectively with medical departments to create a kind of a holistic approach to medicine, something that was quite rare in those days. And as luck would have it, in several years, why the possibility for doing that was really opened by the studies of Boyer and Cohen. And recombinant DNA then became an infectious agent in the department. Everybody was enthusiastic about it, and in that context, we developed shortly in the department individuals with specialties in various aspects of synthesis and use of recombinant DNA. And because one of my previous interests had been in, still was, interest in the development of the pancreas, especially the endocrine pancreas, and insulin was sharply in focus. I became involved early in the cloning of the insulin gene. And during the cloning process...
that was a great day, by the way. Wally Gilbert was giving a major talk at Berkeley the day that Bill announced that he had beaten Wally in the cloning of the insulin gene. That was really awesome. Sorry to interrupt you, Bill.
Once in a lifetime with Wally. At any rate, at the end of this process, why the NIH was going through a process of deciding whether they should legalize, present a law, or develop a process for dealing with this technology, which is considered to be extremely dangerous. And as it turned out, during the cloning process, the regulatory apparatus was not in full function at the NIH, and we got into a situation where the rules were changing at the same time we were doing the cloning. As a result, why we got called in front of a Senate investigating committee with Adlai Stevenson playing the role of the investigator and Senator Schmidt. And the story was basically, well, what happened? Was it dangerous? Was it not dangerous? And Margaret Mead, who was one of the people who testified in that meeting came in with a large staff, like a shepherd's staff. She would pound on the ground and say, these scientists are telling you this is not dangerous. I'm telling you that it's totally dangerous. They don't know what they're doing. They're taking the risk of humanity in their hands. And at that time, the NIH, which was truly supporting our position up until the time that Margaret Mead spoke, left the meeting shortly afterward, and we were castigated for doing this dangerous work. So the net of it was that we became quite interested in not just the cloning of insulin, but in demonstrating safety over risk. And one of the possibilities for demonstrating this was a vaccine. The particular vaccine in mind, hepatitis B, turned out to be technically possible because the genome was so small, 2,700 bases. And at the same time, why I'd been aware of the potential of hepatitis B because I'd consulted for Abbott Laboratories for a long time since my early days, and they were making a lot of money in diagnostics associated with hepatitis B. So I became familiar with the worldwide characteristics, and particularly in Asian people and in China. And so I happened to call Roy Vagelos to discuss with him, first of all, insulin, what should be done with insulin, since Lilly was going to carry out a contract with Genentech, of course. And during the conversation, we were talking about the role of recombinant DNA, and Roy mentioned at the time that they were doing a vaccine for hepatitis B. The vaccine was really based on the isolation of these non-infectious hepatitis B particles called Dane particles, which exist in patients. So it turned out that their vaccine was based on harvesting blood from infected patients, purifying the so-called non-infectious particles and using those. And they were quite far along on this process. It became immediately apparent that it was a self-limiting strategy anyway, but there might be a possibility of doing, performing a mimic of hepatitis B, forming that same non-infectious particle, and in that context form the basis for a vaccine. We got quite excited about this, and afterwards I came home and talked to Pablo about setting up a program with hepatitis B. And Pablo, now take over and talk about this and your experiences with the Merck group.
Yeah, hepatitis B actually was, we were looking for a big virus to sequence at that time. We were very lucky to have the ability to receive from Wally Gilbert's lab the method for sequencing DNA using a chemical approach. That was not published, that was starting to be done, and we became we actually improved the method a little bit because it was a chemical method that depends very much in several reagents that will cut after a guanine and a cytosine, a thymidine, and so on. Suddenly we realized we had the possibility to sequence something important. And so the conversation that Bill had with Merck, you know, the big guys at Merck, Roy Vagelos and Ralph Hirschman, were very much for this project. The lower people at Merck believed they had a vaccine already from infected serum, which was the most crazy thing you can think of, you know. And so our collaborators at Merck didn't believe in us. However, using this method, we decided to sequence the genome of this virus. We had to wait about 6 months before we could clone it because we were in the middle of the moratorium for recombinant DNA. And you realize this was a very effective and dangerous potentially dangerous DNA. We have to sign thousands of little papers before enter to the lab and then change clothes and then go to the next lab to work with this thing, I remember. And now, sequencing DNA wasn't very popular in your lab, Bill. No postdoc, no grad student wanted to do it. So the only way I could push this project was to invite 2 Chilean scientists to join. And I got a little piece of Bill's lab to do it. And Josefina Saldivar, a biochemist from Chile, came, and Margarita Quiroga came, and we set up a sequencing lab, and it took us a year to sequence the 3,000 base pairs of this genome. 20 bases a day, 20 hard-working, with big gels, using a lot of P32 radioactive, like crazy. Thanks to these 2 women, we finally sequenced the genome, and basically we're able to find the structure of the antigen, the main antigen of the surface antigen, for which there was a lot of people trying to get that sequence for a long time. We were able to get it all. That allows us to beat Genentech, and that allows us to beat Wally Gilbert also, because Wally Gilbert was doing exactly the same. The good thing that we beat Wally was using his method for him, and he got the Nobel Prize for that method, the sequences of DNA using the chemical reactions. I think that was very big, having the possibility now of knowing what were the genes and the structure of the surface antigen gene. Then came the second key thing that we had in Bill's lab. We were experts on Saccharomyces, the budding yeast. We did a lot of work with RNA polymerases, isolated the 3 RNA polymerases from yeast, basically work out the whole collection of proteins that these very complex enzymes had from yeast. We continue to work in yeast, isolating genes to study, for example, RNA polymerase 3, we needed the tRNA genes. We isolated the tRNA genes, and for the first time, we sequenced the genes of the tRNA genes and found the intervening sequences. At that time, the only proof of intervening sequences were electron microscopy experiments with loop between DNA and RNA. We actually were the first to determine the exact sequence and location of the intervening sequences. So that was a good work we did also in yeast. So when the question came, where we're going to express the surface antigen, yeast was a possibility that we had all in mind. And that was a key one, because Genentech preferred to use mammalian cells, Art Levinson, and also they started with yeast a little bit late, and the group at Harvard and Biogen decided to do it in E. coli. There was no way that E. coli will produce a particle that needed an animal cell to be produced. And yeast was sort of almost an animal cell, a eukaryote, It was very animal, but very good.
So all of this work happened at UCSF.
It happened at UCSF until we found the particle made in yeast. The most incredible thing is that this particle is a complex structure. This is the first time that you use recombinant DNA to produce just not a protein. This particle has 50 different molecules, different phospholipids, different alcohols, pieces of membrane, and it all was done in the yeast cell. So that was the key that really made the beginning, at least, of this vaccine. But then we couldn't finish at the UCSF. We had to move it to Cairo.
So, Bill, that was the main reason you, the urgency of starting a company, I guess.
Well, fundamentally, the reason we got where we were that we had Pablo and other people in the lab who urgently worked on projects with passion and with efficiency. And let me just point out that in the case of hepatitis B, I talked to Roy at at a meeting and decided, well, this might be a good thing to work on. I came back and I talked to Pablo, and this was on Friday or the weekend. On Monday, we were already started to work on hepatitis B. It was just that efficient a process. But fast forward, this project with Merck was supported by a contract, as was our work on insulin, one from Merck, one from Lilly. And I went to a meeting in Washington set up by Battelle, and I found not just Wally, but several other organizations working on hepatitis B and on insulin, and they were setting up companies. And at the time that this was going on, I personally was very pleased with being on the scientific advisory board of Amgen, and I was really enjoying my life down there. But it became immediately apparent to me that we didn't have the horses to pull off the hepatitis B particle work and the vaccine work by ourselves. And in fact, at the end of that meeting, I called Roy on the phone, Roy Vagelos, and said, asked him if I could come down to Rahway. I just wanted to chat about the current status of affairs on hepatitis B. And we got together, and I explained to him that on the one hand, Amgen was trying to recruit Pablo and Ed too, independently as director of research. Everybody who was really talented had a place to go at a higher salary. I made some efforts at UCSF to try to set up an institute for translation of recombinant DNA technology locally. That turned out to be a non-starter for obvious reasons. And so coming back to this conversation with Roy, after 2 or 3 hours talking over the matter, he said, well, great. If you want to set up a company, we'll set up a company, and we'll give you a contract with the company. And we looked for a place, and fundamentally, in that context, Ed and I and Pablo convinced ourselves that this is a program that we could and ought to do. And in a short period of time, over Easter holidays in 1981, we wrote a business plan. And
No, we thought it was a business plan.
It was a terrible one.
yes, a facsimile of the business plan. And we started.
Yeah. I think there are 4 issues that were very important how Chiron was born. First, we got a very well-established scientific leadership. I'm not going to be one, but those 2 guys here. And we had a lot of scientists that will know us and know our science, and we're doing very good science at UCSF. So we had a group of people that were waiting for us to set up a company and they will join. We are talking of people from Bruce Albert's lab, Ray Lindbergh, Larry Cousins, people from Bill's lab, Patty Tickham, Graham Bell, Leslie Roll, Bob Bishop, Frank Mashar, people from Dan Santi lab were talking to me, Phil Barr, people from, John Sadat, George Kuo. I talked to George Kuo. George Kuo said immediately, I'll work, and so on. And so Ed, some people from Berkeley joined, like Dino Dina, like Bob Quadruna, that was very important at the beginning of Chiron. Steve Rosenberg, and also Tony Brake. So we had this group of excellent scientists that never thought to start a company. They were going academics. But suddenly, because we started this thing and started to talk about the possibility of start a company, suddenly, without any recruiting, we had 20 people there. 20 scientists ready to go. So that's one thing. The leadership, and we had a project, a high visibility project that wasn't done like the hepatitis B vaccine that we had to be finished. And fourth, we had a place. We were already looking for a place, and we have Emeryville already, you know, at 3 cents the square foot. We were,
That's a true story.
and that's
And
how
we actually started inside
and
Cetus.
that's how we were near Cetus.
Right.
And eventually we moved to the Cetus building, because at that time Cetus was in front of us. There were 2 or 3 companies in your building, and there were about 7 companies in our building. Slowly, we got rid of them.
Got rid of the buildings, too.
But anyway,
that
that's
was a great
so
start.
important to start Chiron.
Interesting parallel, because Cetus was started because Don Glaser couldn't get his project done in the university, and to a great degree Chiron was started because Bill couldn't get his projects done at the university. So the necessity to actually go outside and start a company was a common element to both companies. So we did start the company with, anybody want to guess how much our Series A financing was? $1 million. Almost unheard of in today, well, really unheard of in today's world, for sure.
We were underfunded.
But we
Well,
were
that
under-
was the only financing we had before.
Yeah, well, we did have the Merck contract. So we actually had money coming in from a contract when we started. So Bill, moving along then, we had a number of strategies that you especially developed. One was the so-called tripartite strategy, the whole strategy of doing business relationship. Frank alluded that to a little bit as a way to actually find funding for the molecular oncology program at Cetus, but maybe can you spend a minute to describe both that and the emergence of the 50-50 deal, which...
Well, basically, the general idea was that our core strength was research, and what could come out of discovery. First of all, diagnostics with the elaboration of information on a target. And I'd had some experience at Abbott Laboratories as a consultant. I knew something about diagnostics, and I was enthusiastic about it. Of course, with vaccines and with the possibility of mimicry, we became very enthusiastic about being able to mimic infectious agents, and without real mortal danger, as had occurred before with polio, for example, we could, in fact, start a vaccine business and then therapy. So the general idea is the core research could actually provide avenues for commercial development of the 3 different areas. And in that context, we obviously couldn't develop them ourselves, nor did we have the experience to do a fully integrated company, as many companies tried to.
I was the chief financial officer when we went public in '83, and the bankers gave me a book called Accounting Made Simple at the end of our roadshow. So that's an understatement that we didn't have the capability to commercialize these things.
Well, and until Dennis Winger, he was the best financial officer we ever had. So anyway, at that time, the strategy was, okay, our core strength is research. Let's find partners whose core strength is marketing and selling. And have we got a deal for you? It's 50-50. We do the science, we accept your money, and we're going to split the profits 50-50. And okay, in order for that to work, we had to have a compelling argument that the target was important enough to their company, and we were efficient enough so that we could make that idea stick. And as you can imagine, there were very few 50-50 deals done in those days, probably none. because every place we would go, we would get some kind of an equity split. We'd get down to 51-49. Nobody would do 50-50. So fundamentally, we just stayed at 50-50. And sooner or later,
Good work.
we got two 50-50 deals. One was with Ciba-Geigy, and the other was with J&J. J&J was interested in actually diagnostics and growth factors, and Ciba-Geigy at that time was interested in vaccines. And they became, well, first of all, Ciba-Geigy had made the corporate decision not to get into vaccines. So the first thing we had to do was to convince Ciba-Geigy that these are really not traditional vaccines. These are not vaccines. But
Might look like
So-
a vaccine, perhaps not.
we named the company Bioscenes because it's a biological approach to vaccines and it's stuck. And so we formed the Bioscene group with Ciba-Geigy. And that was a terrific relationship run by a fellow named Jakob Nusch, who later became president of the Federal Institute of Technology in Switzerland, fabulous guy. And that began then the process of evolution to the vaccine business. Well, it became apparent after hepatitis B that the vaccine business was complicated and simple mimicry was not going to work in every case. Even now, it's a problem. So there was a strategist at Ciba-Geigy named Richard Williams who thought, well, we have a business, let's start the business at the same time we're developing the new technology vaccines. And the business then was essentially to modernize the old vaccine technology. And it turned out in those times that every country, save the United States, had a corporate organization that produced vaccines for children. So Germany had such a program and started actually from the time of Pasteur, and the same was true for Italy. And we started this, we actually started our program by trying to buy a vaccine company in Canada. Once Ciba-Geigy had committed to the concept of developing vaccines, they were willing to put some real money behind it. So the deal we had in Canada, we're going to buy the Canadian government vaccine organization. The deal we had was that they would put up 95% and we would put up 5% of the money and we would split. And then with profits, in the future, we'd pay back our 45%. So there was a long-term debt that was associated with this. And we got in a bidding war actually with Mérieux, the Mérieux organization in France, which was one of the best known vaccine companies in the world. And the bidding went up and up and up, and it got close to $1 billion Canadian dollars, which is real money in those days.
Still.
Why we decided, on the Chiron side, we didn't want to go forward with this. This was not a deal I wanted to do. And we left Mérieux hanging with a high bid. And they, as a result of this, had to sell the vaccine business later on to Rhône-Poulenc and now Sanofi. And on the other hand, that wedded Ciba-Geigy's interest in vaccines, and we then began to negotiate for the purchase of the vaccine business in Germany. and the one in Italy, Sclavo. And then those two organizations formed an operational unit, which gave us a market entry to a field we wanted to get into, and they supported that supported the research that we were doing in-house on vaccinology, the first targets.
Eventually grew into a $2 billion a year business, which is about what it is now.
That's right.
So the other key element in the Chiron history is the discovery of hepatitis C, Pablo.
Yeah,
Maybe
I
you
think
could talk
I
briefly
think
about how that all
basically
came
what
about.
really made Chiron was the hepatitis C, because it made us a big entry in infectious disease in the blood screening arena. Our decision, and that was a very important, very early decision, and that was 1981 decision, the same year we started Chiron, to continue the hepatitis field and go behind non-A, non-B. Non-A, non-B was what everybody failed in this country for 15 years before we started. And analysts will tell, you are crazy guys. You start a company working in the most difficult and risky project that you can think of. So I think that was a key decision. Very risky, but also very rewarding. We started 81, 82 with hepatitis C. We set up a group. We had close attention to this group. We managed this group we put a lot of effort. Most of Chiron scientists were involved, either sequencing, expressing proteins in yeast, doing the cloning itself of the hepatitis C virus, at that time the non-A agent. And after 5 or 6 years of work, which is a very risky one because it's either yes or no, we find it or we don't find it.
Or somebody else finds it.
Or somebody else. Yes. Actually, we didn't have a lot of competition. The truth, that project will scare everybody, even ourselves. We got it, and we were able to build, a blood screening business. Because we were successful with hepatitis C, we were the first also together with Genentech almost the same time. We sequenced the whole AIDS virus. Having the yeast system, we were able to produce the hepatitis C antigens and the AIDS antigens also, and had this deal with Johnson & Johnson. The government didn't allow us to have a deal with one. We did it also with Apple. We even made a test for the blood of the Hirohito emperor at that time, you see. So basically, why the world blood supply is safe today? It's safe because of our work on hepatitis B, in the AIDS virus, and in hepatitis C. I will say that most of the tests that are being run today in the blood banks, even for AIDS, which we didn't discover, and the hepatitis C for sure, are exactly the same test, because we are not using PCR today for blood screen. Mm-hmm. Still we are using antibodies or antigen, the most traditional test. So I think the impact in worldwide medicine today of the work on infectious disease, it's great. And that came out of that big, bold decision. Without hepatitis C, we wouldn't have been able to do this.
So hepatitis C transformed Chiron.
Absolutely.
Both in many different ways, but especially financially. So in one way, that led up to the merger with Cetus. So Hollings, maybe you can spend a little time telling us about what was going on in Cetus in the late 80s and what the company made the decision, irrespective of Chiron, to put itself on the market at some point in time.
Well, not quite.
Yeah.
Where we were in 1990 was we had put a lot of effort into getting IL-2 to the market, Proleukin. And the first go-around there didn't go so well with the FDA. We didn't. Is that an understatement?
Very badly.
We needed another trial, and that was going to take more capital, and we already had a pretty big organization. And that's actually when I took over the company at that stage and promoted Frank to head of research and made some other changes. We obviously didn't have the kind of money to keep going the way we were, so we made a dramatic scale back in the company. But what we did decide to do was we couldn't raise money at the prices that our shares were at that stage without a big dilution. So we actually made the decision that we were going to monetize PCR because at that stage, we had already basically out-licensed it. And so the $330 million was going to come in pretty handy to recreate the company. And essentially, we're going to recreate it with Frank and molecular oncology and Onyx. Essentially, that was our goal at a loan plan, was essentially to skinny it down even further and to be more of an oncology-specific and specifically molecular oncology-specific company.
That's a widely misunderstood aspect of Cetus. Many people believe you sold PCR for $300 million. The license was already done before that.
Right,
And
yeah,
so that's a very important distinction. It wasn't sold for $300
Yeah, that's what I
million.
meant when I said maybe
Yeah.
if you could rewrite history with the benefit of hindsight, we probably would have tried to do something differently with Roche way back when and maybe held on to it or done something different, but it would have been a dramatic change in the vision of the company at that stage. So without that, we basically said, let's monetize PCR. Let's essentially go with oncology and more molecular oncology and go it alone. And that's actually, I guess, Ed and I had a couple conversations before about combining our efforts in manufacturing, because here we were in Emeryville. We'd built the $30 million plant to produce IL-2 and beta interferon, and you guys wanted to build a plant. And I said, that's nonsense, Let's do it together. We'll figure out how to share the cost and whatnot.
There was a bridge across the street in Emeryville, still is, I think.
Still there.
That was
Well, the
Cetus
building's
on
gone,
one
I think,
side and
before.
Chiron. Oh, the
Building's
building
gone.
is gone
Building's
now,
gone,
that's
yeah.
right, been taken down. The doors were locked on both ends, but if you had,
But fundamentally, I think we got to a position where we had to go to loan plan, but then Bill and Ron Kate began a discussion about maybe combining the companies. We really weren't putting the company up for sale. I think we were looking for options, but we could solve it with really the sale of PCR, which we were going to do in any event. And it was okay with Chiron that we sell it because they had branch DNA. So not only did Onyx let it get away for $330, but Chiron let it get away for $330 million.
that's also true.
So anyway, the bottom line, though, is the merger came together. I think it was a very successful merger. We did have this bridge that we could easily
Of instruments through the bridge.
yeah. But the integration, I think, was very successful. We kept a lot of the skills that were at Cetus probably had more of the biologic manufacturing and capabilities and some of the regulatory stuff to get the drugs approved. And so those teams stayed in place to get both beta interferon and IL-2 approved. I'd say my big failure when I joined you two guys in the office of the chairman was I let that guy at the end get away. And
Yes.
It was that bad, huh, Frank? Yeah.
Well, the other, if I can just chip in there a little bit, the other thing was that 120 of the scientists who were in the PCR group working for John Snitsky over here actually then joined the Roche organization to
Yeah,
commercialize
you had left
the thing.
before that and went
I'd
to
left
Roche, Tom.
3 years before that, so basically everybody came back, and I was head of R&D at Roche, with John head of research. So,
But I
worked
think
out
that's
really
a pretty
well,
interesting
terrifically,
model,
yeah.
really, for how to do things, because Cetus did what it did best in PCR, which was the science. And then Roche did what it did best, which was to develop the commercial side of the whole business, regulatory and commercial side of the whole thing. So that worked really well. But coming back to the merger, I'd say, again, we would have done Onyx on our own within. I think probably that was in your eyes, Bill, at least, to say that was a program that you wanted to keep. But Frank was, these guys here at the front row, these VCs, were whispering in Frank's ears, not these particular guys. They weren't the culprits, but it was the VCs who basically talked Frank into saying, hey, look, you could build your own company. Why do you need Chiron's money? In fact, you had to write a plan for Bill, right? It was called Chiron-cology, I think it was.
That's what it's called, yeah. Yeah.
It turned out to be the business plan for Onyx in the end. So anyway, the story, Frank can tell the story, and others, me and others, later about Onyx, but I think the merger, the cultures were close, the skill sets balanced one another. We probably had a little bit more on the therapeutic side. You had the vaccines and the diagnostics side of things. And it seemed like the integration went about as well as you can expect. Two large, at that stage, two pretty large biotech companies. They're about 600 people each, I think, something like that.
Yeah.
So it was a lot of fun merging the two companies.
Well, I think the scientific rationale was largely that there was a growing synergy between oncology and infectious disease, which I think has only been enhanced in the days since then.
Yes.
So Bill, you...
And also the idea was that both companies needed to grow to become competitive in this arena. And proximity, and there was a lot of friendship across the street, made it likely that if we were going to do anything like this was the time to do it. We were lucky enough, after conversations with Ron Cape, then to get Fred Frank involved, who represented Ron, and our own attorneys. And Fred knew very well Fritz Gerber at Roche. So there was the issue of how to pull the deal off, because eventually it turned out to be an $800 million deal. We didn't have that kind of capital. But we were able to sell then the PCR rights that Roche had. We were able to sell the rest of those rights to Roche for $200 million. And that plus the equity part of the transaction made it work. So there was a lot of goodwill on all sides and some, I'd say, brilliant banking done by particularly Fred Frank in this case.
Now
And
works
Fritz
with
Gerber
Steve.
had been a friend even before he started Genentech. And so he helped facilitate this deal as well. A lot of goodwill on all sides.
The worst part, we had to lay off a lot of people to make the merger work. We had way too many people, the two companies. One of the worst I can remember, one of my closest friends' wife worked at Chiron. She was on the list, didn't do a lot for our friendship. But it's really hard to go through a massive layoff during a time of actually good things happening, because people just couldn't understand, and rightfully so. Gee, things are going well, why did I lose my job? It was a very difficult thing. In terms of lessons we all learned, for me, that was one of the most painful experiences of the Chiron, and I'm sure Cetus history, was the reality that we had to downsize the organization for people from both sides in order to right-size it for us going forward.
But I think if you kind of look back at the history of these two companies and you say what really happened here, it's the best science. Really, the story is the best science that you could possibly have. And I think that's still the message, I think, for this industry, is even though we don't have the low-hanging fruit of some of the things that we worked on, it's more complicated today, but we're just still at the edge of understanding human disease. And we got great tools now to work on PCR and a lot of other things that actually allow us to understand what's going on in human disease and intervene.
Yeah.
We don't know what targets necessarily to work on, so it's tougher. I think that's clearly one of the lessons for me, anyway. And I think the other lesson for me is really focusing. As a small company, focus in what you do best. Don't try to do everything. I think there were times when we tried to do too many things in the Cetus days, for sure. Yeah. And applying it kind of to the Onyx, we adopted a vision there to say, we're going to just basically change the way cancer is treated. That was our vision, and we did nothing other than that. And so we felt like we were always the best at that, and that was sort of, I think, a lesson that came out of where we had been in the Cetus days, where we were trying to do a lot of things, that maybe too many things that didn't differentiate ourselves.
Yeah. Well, as we say, it was a different era. I mean, the early days of Cetus were fuels and agriculture products, et cetera. The early days of Amgen, as two of our guests here remember well, indigo dyes and also fuels, et cetera, because the technology was broadly applicable.
Right.
And it took a long time for all of us to figure out that the technology is not a business. It's a technology, and the businesses are pharmaceuticals or energy or whatever, the various things. And so a lot of focusing had to occur, but certainly in today's world, you could never start a company that looks anything like these. But the element, I think, still distinguishes good companies from bad is having real scientific horsepower.
Right.
And in light of today's conversations at the Supreme Court about gene patenting, without intellectual property, none of us would be sitting here today. We'd all be doing something else. Only the reality of intellectual property allowed us to monetize our assets. Our
And we
hosts are
enjoy,
getting nervous about the time.
yeah, we enjoy a very, we should say also, we enjoy a very good internal company in the intellectual property. And Bob Blackburn is here today. We got an incredible position on hepatitis. We got a lot of potential other inventors and people attacking that patent. And we got an excellent position that even today is quite an achievement. And we had
Similarly on the Cetus side for a number
Yes.
of things that happened,
Yeah.
and including PCR.
So that was very important.
Yeah.
Well, it's a lot tougher for newer companies today, too, because, I mean, we had the benefit of capital markets in the early days. That, you know, when we raised 110, Steve, what was it, 110, 108, 108.5 million, the largest offering at the time of any public company, even Ford Motor Company when they went public. You know, we basically had dreams. We had proteins that hadn't yet been shown to do anything at
Bubbles.
that stage. And we had this wide variety of different things. But the capital markets are very selective today. It's very difficult for young companies. So you got totally different business models than the ones that we were able to basically stumble through at some level to ultimately create the company. So.
If I could make two more points, I think there still is this element of risk in everything, because it's 25 years since the discovery of hepatitis C and there's still no vaccine, even though
Yep.
it's logical to have the vaccine business. There's 30 years
No
since
AIDS
HIV, no vaccine. So it's like TNF turned out to be a terrible drug, but the opposite of it turned out to be a great drug. And the other thing is, I think target validation, given the status of genomics, has transformed how targets will actually reach a stage where companies can actually then do drug development. But the drug development still has to happen at companies, not in academia or any institute or anything. That is a ridiculous concept. It's not going to work.
Well, our hosts are getting antsy, so I think
Yes.
we'll have wrap up. Anybody want to make any closing comments? No, one final, it's amazing to me how evolved people able to traffic now between academia, small business, large business, and you know, in another era, if you left academia, you were gone forever. They kissed you goodbye and didn't want you. In today's world, I mean, Frank's a good example. Has been in and out of companies and in academia. And
Well,
Tom,
in one company and started another one and then back to academia.
yeah, and Tom was in Cetus and then a big company, Roche, and now he's a Regents lecturer at UC Berkeley. An amazing, wonderful mix, I think, of the way people lead their lives, which is very different than it used to be. Bill, do you want to make any closing comments? Frank?
Our motto at Cetus was, we will lead biotechnology wherever it takes us.
Bill, do you have any closing comments you'd like to make?
Well, it was probably an experience that had a great influence on everybody who worked with us in that wonderful organization. I mean, we didn't talk about the terminal parts of Chiron. The last step of which, or the first step of the last process, was the purchase by Ciba-Geigy of 49% of Chiron. And that came about because of the great capital needs for growing the business in each one of these areas. Because of our strength in diagnostics, it turned out that Ciba-Geigy had a diagnostic business. So the proprietary position we had in diagnostics was so strong with HIV, HBV, HCV, that trio, that nearly every other diagnostic company, if they were a broad-scale company, had to deal with that intellectual property and the position in the marketplace. So essentially, Ciba's own diagnostic company was devalued, and therefore, in some senses, the best place for it to be would be with Chiron. Similarly, the power in the vaccine strategy was at Chiron because we were running the 50-50 joint venture at the time. So that led then to a proposal that they partner with Chiron on a part basis, sort of a 50-50 basis, providing capital, their half of the vaccine business, their total diagnostic business, to several $100 million, and about $1 billion in cash in different forms. So eventually it turned out to be a deal that was it was impossible to turn down, and we embraced it and with great enthusiasm. And then it turned out that CEVA merged to form Novartis, and in that context, the structure and mission of CEVA changed significantly. And after that, then the relationship between the companies began to slowly dissolve. In the context, there was just not a close relationship as was the case with Roche and Genentech. And that led then to a series of events which culminated in the purchase of the company by Novartis.
Well,
That's the story.
anybody else want to make a closing comment? Not?
Chiron's name is known internationally. I was in Cambodia in February, and as we were walking down the street, and a guy rode by on a motorbike with a Chiron T-shirt on.
Well, there are a lot of T-shirts around. But one of them was the merger T-shirt, and too bad somebody in the audience didn't bring it. It was a cute shirt with the whale, and that I think Larry Kurtz designed, if I remember correctly. Yeah, well, yeah, great.
Should have worn it.
Well, thanks to all of you for listening to us go on. We're happy we're still alive and able to share this with you. And it's,
Always still remember.
We're still leading.
So let me just make a couple of comments in closing. 1953, Watson and Crick got at the structure of DNA. It was roughly 20 years later, the early 70s, that these companies got started. It was another 20 years, early 90s, that the industry that we then created became profitable. And now we're 20 years later, 60 years from Watson and Crick. There are now over 10,000 biotech companies in the world today. We have hundreds of products that are in the market today as a result of what we all did in building this industry a long time ago. Interestingly enough, we crossed the $1 trillion mark this year in terms of capital that we raised in this industry. Wow. That capital we raised to finance the dreams and hopes of guys just like this. It's absolutely amazing. Today, we are still raising capital. We, the industry, raised some $70 billion last year. We were raising $1 million at a time a long time ago. We created along the way an enormous array of financing tools. We created R&D limited partnerships, and we created some fancy accounting and tax maneuvers. We created junior stock. The industry was as creative in its ability to attract capital as it was in its ability to move science. And so on behalf of all of us, we want to thank these pioneers for what they did and what they continue to do to build this incredible industry. So Ed, for your inspired leadership of the panel, and Frank and Hollings and Bill and Pablo and Tom, thank you very much for doing this. I also want to just quickly thank the promotional partners for this. We had some support from Bay Bio, from UCSF, from QB3, obviously from Ed and Hollings and Bill. We have a tremendous amount that we want to thank all of these. It's a good story. It needs to be told, and we're just honored that we could reflect on it tonight. So thank you very much for coming. We have a reception, I think, in the back of the room, and continue to support the activities of these individuals and the foundation that's trying to make sure we've recorded the history accurately. Thank you very much.
Yeah. Thanks.